Malaria. Before cities rose from dust, before empires carved their names into stone, before humanity dared to dream of conquering the world, it was already there, waiting. A single-celled assassin forged in the crucible of evolution, perfected across millions of years to do one thing: survive inside us. It rode the bloodstreams of our ancestors out of Africa. It toppled Roman legions. It devoured armies. It rewrote the genetic code of entire populations, more deadly than every plague, every war, every catastrophe in human history combined. Invisible, unstoppable, inevitable, one mosquito bite, one parasite, and the human body becomes a battlefield where billions wage war in silence. Tonight, a child will burn with fever. Tomorrow, hundreds of thousands will fall. This is not just a disease. This is the ultimate predator. This is humanity's oldest enemy. This is malaria. Before we begin, if you enjoy these types of interesting science topics, hit the like button, comment, and tell us where you are listening from. If you'd like to hear more stories like this, you know what to do. Simply subscribe. Deep in the equatorial forests of Central Africa, where sunlight filters through layered canopies and water pools in the shadows of ancient trees, a microscopic partnership was forged millions of years before humans walked upright. The story of malaria begins not with people, but with parasites older than our species, single-celled organisms belonging to the genus Plasmodium, drifting through evolutionary time in the bloodstreams of great apes. In these primordial forests, mosquitoes have always been architects of connection, drawing blood from one creature and carrying invisible cargo to another. The Anopheles mosquito, a slender insect with spotted wings and a distinctive angled posture when feeding, became the vessel for Plasmodium parasites that had already learned to exploit the warm, nutrient-rich environment inside primate bodies. Long before the first humans stood upright on the savanna, these parasites moved between gorillas and chimpanzees, refining their strategies of invasion, replication, and transmission across countless generations. The forests of what we now call Central Africa were laboratories of coevolution. Here, in the permanent twilight beneath dense vegetation, where standing water collected in tree hollows and forest floor depressions, mosquito larvae wriggled and transformed. Adult females emerged hungry for the blood meals necessary to nourish their eggs. When they fed on infected primates, they became more than predators. They became carriers, hosts to parasites undergoing their own complex metamorphosis within the insect's gut and salivary glands. Our hominin ancestors, moving through these same forests several million years ago, entered an ecosystem already saturated with malaria. The earliest humans, gathering fruit and hunting small game, were bitten by the same mosquitoes that fed on other primates. Initially, the parasites that infected apes may have caused little more than transient illness in humans, their biological machinery not yet perfectly calibrated to our physiology. But evolutionary time is patient and relentless. As human populations grew and diversified, so too did the parasites. Through countless transmission cycles, through innumerable hosts and innumerable mosquitoes, certain Plasmodium strains began to specialize. They adapted to human blood chemistry, to human immune responses, to the particular temperature and environment of human bodies. What emerged were parasites exquisitely tuned to exploit humans as their primary hosts, particularly Plasmodium falciparum, the most deadly of the malaria parasites, which appears to have crossed from gorillas to humans, perhaps 50 to 100,000 years ago. The relationship between parasite, mosquito, and human formed a triangle of dependence. The Plasmodium parasite cannot complete its life cycle without both a mosquito and a vertebrate host. The mosquito provides the space for sexual reproduction of the parasite, while the human body offers explosive asexual multiplication. Neither can the parasite survive long without the other. It is an obligate partnership, locked together by evolutionary necessity. When early humans began to leave Africa, Moving northward and eastward into new climates and new continents, they did not travel alone. In their blood, invisible and patient, traveled the parasites, and wherever humans settled near water, wherever they created the conditions mosquitoes needed to breed, malaria followed. It was a shadow pandemic, operating beneath the threshold of historical record, shaping human settlement patterns in ways we are only beginning to understand. The forests where malaria originated still exist, though diminished and fragmented. The mosquitoes still breed in forest pools. The parasites still circulate among primates. And in rural villages at the forest's edge, where humans and mosquitoes share close quarters, the ancient transmission continues. The disease that emerged from these forests has become one of humanity's most persistent companions, a reminder that we remain part of an ecosystem far larger and older than civilization itself. Understanding malaria's origins requires stepping back to see not just a disease, but a relationship, one forged in evolutionary time, shaped by ecology, maintained by biology. The parasite did not choose humans. We entered its world. And once we were there, once the transmission cycle was established, once human populations grew large and dense enough to sustain ongoing transmission, there was no easy escape. The forests of Central Africa gave birth to a global phenomenon that would follow humanity through every age, every migration, every attempt to tame the natural world. The infection begins with a bite, a brief moment of contact so commonplace in tropical regions that it often goes unnoticed. A female Anopheles mosquito, guided by carbon dioxide from human breath and heat radiating from skin, lands on exposed flesh. She is not feeding for sustenance but for reproduction. Blood provides the proteins necessary to develop her eggs. What makes this particular bite dangerous is invisible. The mosquito is already infected, carrying in her salivary glands thousands of microscopic parasites in a form called sporozoites. As the mosquito's proboscis pierces skin and probes for a blood vessel, she injects saliva containing anticoagulants to keep blood flowing. Mixed with that saliva are the sporozoites, elongated, motile cells that immediately enter the bloodstream. Within minutes, they are carried through the circulatory system, flowing through arteries and capillaries, searching for their first destination, the human liver. The liver is the malaria parasite's hidden sanctuary, a biological fortress where the initial invasion unfolds in secret. Sporozoites pass through liver sinusoids, specialized capillaries with wider gaps between cells, and invade hepatocytes, the primary cells of liver tissue. Each sporozoite that successfully enters a hepatocyte has reached temporary safety, hidden from immune surveillance in the body's largest internal organ. Inside liver cells, the parasites undergo a profound transformation. Each single sporozoite begins to multiply, not through ordinary cell division, but through a process of rapid, repeated nuclear division, followed by segmentation, like one cell becoming many without the usual intermediate steps. Over the course of roughly a week to 10 days, depending on the Plasmodium species, one sporozoite becomes tens of thousands of new parasites. These are merozoites, fundamentally different from their sporozoite parents, adapted for the next phase of invasion. The infected liver cell eventually ruptures, releasing its cargo of merozoites into the bloodstream. This moment marks the transition from the silent, symptom-free liver stage to the active, visible blood stage of infection. The merozoites, now circulating freely, begin seeking their next target: red blood cells. Red blood cells, erythrocytes, are among the body's most abundant cells, numbering in the trillions, constantly circulating, carrying oxygen from lungs to tissues. They are also, paradoxically, among the simplest of human cells, lacking even a nucleus or the usual cellular machinery. For malaria parasites, they represent ideal homes, abundant, nutrient-rich, and relatively defenseless. Merozoites attach to the surface of red blood cells using specialized proteins that fit with target molecules on the cell membrane like biological keys entering locks. The parasite triggers the cell to engulf it, creating a protective bubble of vacuole inside. Once inside, sheltered by the cell's own membrane, the parasite begins to feed on hemoglobin, the oxygen-carrying protein that gives blood its red color. The parasite digests hemoglobin as its primary food source, breaking down the protein and converting the iron-containing heme into dark crystals of hemozoin. It grows, transforms from ring-shaped to more complex forms, eventually filling much of the red blood cell's interior. Then, once again, it multiplies, one parasite becoming 8, 16, or more, depending on the species. The red blood cell, now packed with parasites, ruptures. The new generation of merozoites spills into the bloodstream, each seeking a fresh red blood cell to invade. This cycle invasion, feeding, multiplication, rupture, reinvasion repeats every 2 or 3 days, depending on the parasite species. With each cycle, the number of infected cells can double, then double again. Within weeks, if untreated, millions or billions of parasites can be circulating through the bloodstream, each one destroying red blood cells, each one contributing to the cascade of symptoms and damage. But the parasite's life cycle has another dimension. Some merozoites, instead of continuing the asexual replication cycle, differentiate into sexual forms called gametocytes. These cells do not multiply in the human host. They wait, circulating in the bloodstream, for the chance encounter that will complete the cycle. Another mosquito bite. When a mosquito feeds on an infected person, she draws up blood containing these gametocytes. In the mosquito's gut, the gametocytes transform into male and female gametes that fuse in sexual reproduction, creating new genetic combinations. The resulting zygote becomes mobile, burrowing through the mosquito's gut wall and forming a cyst on the outside of her stomach. Inside this cyst, more rounds of multiplication occur, eventually producing thousands of new sporozoites that migrate to the salivary glands. The mosquito is now infectious, ready to transmit parasites to the next human she bites. This is how malaria forms and begins, not as a single infection but as a complex, multi-stage journey through 2 hosts. The parasite is not simply invading, it is navigating, transforming, adapting to radically different environments: human liver, human blood, mosquito gut, mosquito salivary glands, completing a cycle that neither host alone can break. The elegance and complexity of this life cycle, refined over millions of years, is precisely what makes malaria so difficult to interrupt, so persistent, so successful at maintaining itself in human populations wherever conditions allow. The spread of malaria operates on 2 interconnected scales. Within the human body, where the disease progresses through escalating cycles of parasite multiplication and cell destruction, and between humans, where mosquitoes serve as living syringes, transferring infection from person to person through their feeding patterns. Inside the body, progression follows the rhythm of the parasite's replication cycle. After the initial release from the liver, merozoites invade red blood cells, mature, multiply, and burst free to invade new cells. This happens in rough synchrony, large populations of parasites reaching the rupture stage at approximately the same time. When millions of infected red blood cells burst nearly simultaneously, they release not only new parasites but also cellular debris, waste products, and molecular signals that trigger the immune system. The rupture events create the characteristic fever pattern of malaria. In infections with Plasmodium vivax or Plasmodium ovale, the cycle repeats every 2 days, causing fever spikes at regular intervals, tertian malaria, so named because fever recurs every third day by historical counting methods. Plasmodium malariae follows a 3-day cycle, creating quartan malaria. Plasmodium falciparum, the deadliest species, often lacks the precise synchrony of other types, producing a more continuous or irregular fever pattern. With each replication cycle, the parasite burden increases geometrically. One infected cell becomes 16. 16 become 256. The progression is exponential. In severe, untreated falciparum malaria, parasite density can reach levels where 1 in every 10 red blood cells is infected, even higher in fatal cases. The destruction of red blood cells accelerates, leading to anemia, insufficient oxygen-carrying capacity. The spleen, filtering blood and trying to remove infected cells, becomes enlarged and overworked. The bone marrow attempts to compensate by producing new red blood cells faster, but cannot keep pace with destruction. The parasites also modify infected red blood cells in ways that change their behavior. Falciparum parasites, particularly, cause infected cells to become sticky, adhering to the walls of small blood vessels. This sequestration keeps infected cells out of the spleen's filtering system, protecting parasites from removal, but it also impedes blood flow. In the brain, sequestered cells can block capillaries, restricting oxygen delivery to neural tissue. In the placenta during pregnancy, they accumulate in ways that compromise fetal development. This cellular adhesion transforms a blood infection into an organ-threatening disease. The immune system mounts increasingly aggressive responses as the infection progresses. It recognizes parasite proteins, cellular debris, and chemical signals as foreign threats. Inflammatory molecules flood the bloodstream, triggering fever, chills, sweating, and muscle aches. Immune cells attack infected red blood cells, contributing to anemia even as they attempt to control the infection. In severe cases, the inflammatory response itself becomes dangerous, contributing to capillary leakage, swelling, and organ damage. Between humans, spread requires mosquitoes, and mosquitoes require specific environmental conditions. Female Anopheles mosquitoes lay their eggs in water not flowing streams, but still or slow-moving water where larvae can develop. Rice paddies, irrigation channels, swamps, puddles, containers, and even animal hoof prints filled with water can serve as breeding sites. The larvae hang at the water's surface, breathing through tubes, feeding on microorganisms. Within days to weeks, they pupate and emerge as adults. The geography of transmission maps directly onto the geography of suitable mosquito habitat. Where water is plentiful and temperatures warm, tropical and subtropical regions. Rainy seasons, irrigated agricultural areas, mosquito populations explode. A single female can lay hundreds of eggs in her lifetime. In optimal conditions, populations can increase from negligible to enormous within weeks. Temperature governs every aspect of transmission. The Plasmodium parasite develops inside the mosquito at rates determined by ambient temperature. In cool conditions, development is slow or stops entirely. Malaria transmission generally requires temperatures above 15 to 20 degrees Celsius. In warmer conditions, the parasite completes its mosquito stage development faster, and mosquitoes themselves feed more frequently. The warmest regions near the equator maintain year-round transmission. Farther from the equator, transmission may be seasonal, concentrated in warm, rainy months. Human behavior and living conditions profoundly influence spread. In regions where people sleep outdoors or in homes without window screens or bed nets, mosquitoes have easy access to human blood meals. Homes built on stilts over water, agricultural workers sleeping near irrigated fields, mining operations creating stagnant pools, all create interfaces where humans and mosquitoes meet repeatedly. Population density matters too. In isolated households, transmission chains break easily, But in villages and cities, infectious humans and susceptible humans live close together, allowing mosquitoes to efficiently transfer parasites from person to person. The disease spreads not uniformly, but in patterns shaped by ecology and society. Children, having not yet developed partial immunity through repeated exposure, are infected more frequently and more severely. Adults in highly endemic areas may carry parasites with minimal symptoms, serving as reservoirs that maintain transmission even when severe cases are less visible. Travelers or migrants entering endemic regions without prior exposure face high risk. Agricultural practices, dam construction, deforestation, creating new pools, climate patterns all modulate the intensity and geography of spread. Malaria also spreads through time, persisting in populations through dry seasons when transmission drops, then resurging when rains return and mosquito populations rebound. The parasite can hide in human livers in dormant forms, hypnozoites reactivating months or years later to restart blood-stage infection, ensuring that transmission can resume even after long periods without mosquito activity. The progression within the body and the spread between bodies are not separate phenomena but aspects of a single, integrated biological system. The parasite exploits both human and mosquito physiology, using each host for different purposes, creating a cycle that requires both to continue. Breaking that cycle, stopping either the progression within individuals or the transmission between them, is the fundamental challenge of malaria control. Neither alone is sufficient. The parasite's strategy is redundant, resilient, evolved over millions of years to persist, despite immune responses, seasonal changes, and the countless other obstacles to its survival. Malaria's journey from African forests to global ubiquity is inseparable from the story of human civilization itself. As humans migrated, settled, farmed, traded, and waged war, they carried the infection with them, seeding it into new continents, new ecosystems, new populations. The disease shaped history even as history shaped its spread, creating a feedback loop where human choices and malaria's biology influenced each other across millennia. The earliest civilizations of the Mediterranean world knew malaria intimately, though they lacked the vocabulary to describe it as a single disease. Ancient texts from Greece and Rome describe recurring fevers, enlarged spleens, and devastating illnesses associated with swamps and marshlands. The Roman Empire experienced malaria as an endemic presence, particularly in the countryside surrounding Rome itself, in the Campania region where marshes provided ideal mosquito habitat. Some historians argue that malaria contributed to Rome's eventual decline, weakening rural populations, reducing agricultural productivity, and affecting military recruitment. The disease was so closely associated with marsh environments that the Italians named it malaria, bad air, believing that poisonous vapors rising from swamps caused the illness. This miasma theory, though incorrect in mechanism, captured a real ecological truth. Swamps harbored mosquitoes, and mosquitoes transmitted malaria. The association was consistent enough that even without understanding the actual vector, people recognized the connection between standing water, certain environments, and sickness. In Asia, malaria established itself across vast territories from the Middle East through South Asia to Southeast Asia and southern China. Different Plasmodium species found different niches, vivax thriving in temperate zones, falciparum dominating tropical regions. Agricultural intensification, particularly rice cultivation requiring flooded paddies, created expansive breeding grounds for mosquitoes. Malaria became endemic across rice-growing regions, a predictable part of life that shaped settlement patterns, farming calendars, and social organization. The disease reached the Americas through two routes. Plasmodium vivax may have crossed with early human migrations from Asia, though evidence is debated. But the introduction of Plasmodium falciparum, The deadliest form came definitively with the transatlantic slave trade beginning in the 16th century. Ships carrying enslaved Africans across the Middle Passage also carried infected mosquitoes in water barrels and infected humans in cargo holds. Once in the Americas, falciparum malaria found suitable mosquito vectors already present and exploded across tropical and subtropical regions from Brazil to the Caribbean to the southern reaches of North America. The Columbian Exchange thus became an epidemiological catastrophe in multiple directions. European colonizers and enslaved Africans brought malaria to the Americas while simultaneously encountering yellow fever and other diseases. Native American populations, having no prior exposure to malaria and no evolved genetic adaptations, suffered tremendously. The disease contributed to the demographic collapse of indigenous peoples, compounding the devastation of smallpox, measles, and other introduced infections. During the construction of the Panama Canal in the late 19th and early 20th centuries, malaria and yellow fever killed thousands of workers. The French attempt to build the canal collapsed partly because of the overwhelming disease burden. The American effort succeeded only after implementing mosquito control measures based on newly understood principles of vector transmission, draining standing water, screening living quarters, applying larvicides. This represented one of the first large-scale applications of scientific understanding to control malaria, demonstrating that the disease could be managed through environmental modification and mosquito population reduction. Malaria followed military campaigns and wars throughout history. Armies marching through endemic regions suffered massive losses to disease, often exceeding battle casualties. Alexander the Great may have died of malaria. The disease affected military outcomes in the American Civil War, the First and Second World Wars, and conflicts throughout Asia and Africa. Soldiers sleeping outdoors, moving through tropical jungles, establishing camps near water sources all created ideal conditions for transmission. The middle decades of the 20th century saw humanity's most ambitious attempt to eliminate malaria globally. Following the Second World War, the development of synthetic insecticides, particularly DDT, and effective anti-malarial drugs seemed to offer tools capable of breaking transmission cycles. The World Health Organization launched a global eradication program, spraying homes with insecticides to kill mosquitoes and treating infections with drugs to eliminate the parasite from human populations. The campaign achieved remarkable successes. Malaria was eliminated from Europe, North America, and large parts of Asia and South America. Countries that had experienced endemic transmission for centuries saw cases drop to zero and remain there. In southern Europe, where malaria had been present since ancient times, the disease disappeared entirely. But in tropical Africa, where transmission intensity was highest, where mosquito populations were enormous and diverse, where healthcare infrastructure was weakest, eradication proved impossible. The eradication program faced multiple obstacles. Mosquitoes developed resistance to insecticides, parasites developed resistance to drugs, funding proved insufficient, and the biological intensity of transmission in equatorial Africa exceeded the tool's capacity to interrupt it. By the late 20th century, the goal shifted from global eradication to control, from elimination to burden reduction. Malaria persisted, concentrated increasingly in sub-Saharan Africa and certain regions of Southeast Asia and South America. The late 20th and early 21st centuries brought new challenges. Drug resistance spread, with parasites evolving resistance to chloroquine, then sulfadoxine pyrimethamine, then other antimalarials in succession. Insecticide resistance emerged in mosquito populations. Armed conflicts disrupted control programs. Population growth increased the number of people at risk. Climate patterns shifted mosquito ranges, and yet, progress continued. New drugs, improved diagnostics, wider distribution of insecticide-treated bed nets, better surveillance, and increased funding from international organizations led to substantial reductions in malaria deaths and illness from peaks in the early 2000s onward. The historical geography of malaria demonstrates how deeply intertwined human activity and disease transmission can become. Every dam built, every forest cleared, every irrigation system installed, every war fought, every population movement altered the equation of transmission. Malaria has been both a driver of history, influencing where people could live, which armies won battles, which construction projects succeeded, and a product of history, spreading along trade routes, expanding with agriculture, persisting in regions where poverty limits access to preventive measures. The disease that began in African forests is now concentrated back in Africa, completing a strange historical circle. The regions that eliminated malaria did so through economic development, infrastructure improvements, healthcare investments, and environmental modifications that reduced mosquito populations. The regions where malaria persists most severely are those where poverty, limited healthcare infrastructure, and ecological conditions that favor intense transmission create a self-reinforcing trap. Breaking free requires not just medical interventions but economic development, infrastructure investment, and sustained political commitment, lessons written across centuries of human struggle with this ancient infection. In a village in sub-Saharan Africa, a young child wakes in the night, burning with fever. Her body trembles uncontrollably, despite blankets and the warm tropical air. The shivering lasts an hour, perhaps longer, until suddenly it breaks and fever spikes, skin hot to touch, consciousness clouded, small body consumed by heat. Then, as abruptly as it came, the fever subsides, sweat soaks the bed mat, the child sleeps, exhausted. This pattern may repeat in cycles every 2 or 3 days, or it may progress continuously, blurring into a cascade of overlapping symptoms. This is the face of malaria in the modern world, a disease that kills hundreds of thousands of people annually, the vast majority of them children under 5 years old in sub-Saharan Africa. It is a disease of the poor, of the rural, of those without reliable access to prevention or treatment. Geography, economics, and biology intersect to create patterns of suffering that follow the lines of global inequality with terrible precision. The experience of malaria varies enormously depending on the parasite species, the intensity of transmission in a region, the age and immune status of the infected person, and access to healthcare. In regions of intense year-round transmission, much of equatorial Africa, children experience multiple infections per year from infancy onward. Those who survive early childhood gradually develop partial immunity, enough to control parasite density and prevent severe illness, though not enough to prevent infection entirely. Adults in these regions may carry parasites with minimal symptoms, their immune systems holding the infection in check through constant, exhausting vigilance. In areas of lower or seasonal transmission, regions farther from the equator, highlands with cooler temperatures, areas with strong control programs, people develop less immunity. Infections are less frequent, but potentially more severe, because the immune system has less experience fighting parasites. Adults and children alike may develop dangerous illness from first infections. The initial symptoms are often nonspecific: fever, headache, muscle aches, fatigue, sometimes nausea and vomiting. These could be many diseases. In regions where malaria is common, people may not seek care immediately, assuming the illness will pass. But malaria has a distinctive pattern for those who know to look. The cyclical fevers, the rigor's violent shaking chills followed by drenching sweats, the profound exhaustion that makes even sitting up feel impossible. When infection is caused by Plasmodium falciparum and left untreated, progression to severe disease can happen rapidly, especially in children. pregnant women, and non-immune adults. Severe anemia develops as red blood cells are destroyed faster than the body can replace them. Skin and eyes may take on a pale or yellowish tint. The child becomes lethargic, uninterested in food or play, breathing rapidly as the body tries to compensate for reduced oxygen-carrying capacity. Cerebral malaria represents the most feared complication. Infected red blood cells sequester in brain capillaries, blocking oxygen delivery to neurons and triggering inflammation. The child may have seizures, lose consciousness, slip into a coma. Even with treatment, mortality is significant, and survivors may have lasting neurological damage, learning difficulties, motor problems, cognitive impairments. The transformation from a feverish but conscious child to one in a coma can happen within hours, creating urgency that healthcare systems in rural areas often cannot match. Respiratory distress, difficulty breathing, rapid respirations, fluid accumulating in lungs can develop as inflammatory responses affect pulmonary blood vessels. Kidneys may fail, unable to filter blood properly as circulation is compromised. In pregnant women, malaria causes severe anemia, increases risk of miscarriage, and leads to placental infection that compromises fetal growth. Babies born to mothers infected during pregnancy are often underweight, making them vulnerable to other health problems. The modern geography of malaria is stark. Sub-Saharan Africa carries roughly 95% of global cases and deaths. Nigeria, Democratic Republic of Congo, Tanzania, Mozambique, and Niger account for most cases. Southeast Asia, particularly the Mekong region, and parts of South America, especially the Amazon basin, maintain significant transmission. India experiences the majority of cases outside Africa. But large parts of the world that once had endemic malaria, Europe, North America, much of Asia, are now malaria-free. Access to prevention and treatment creates life or death differences. In well-resourced settings with early diagnosis and appropriate treatment, malaria is rarely fatal. A traveler from a non-endemic country who develops malaria can receive prompt diagnosis, effective drugs, and supportive care, almost always surviving. In rural sub-Saharan Africa, a child may live hours or days away from a health facility, may not have access to rapid diagnostic tests, may receive delayed or inadequate treatment, may face stockouts of essential medications. The same disease, vastly different outcomes. Insecticide-treated bed nets have become the primary prevention method in endemic regions. Sleeping under a net creates a physical barrier between mosquitoes, which feed primarily at night, and sleeping humans. The insecticide kills mosquitoes on contact and repels those that approach. In areas with high net coverage, malaria transmission drops substantially. But nets require consistent use every night, must be replaced periodically, and are less effective when damaged or when people sleep outdoors. Indoor residual spraying, applying insecticides to walls and ceilings where mosquitoes rest after feeding, provides additional protection in some settings. Artemisinin-based combination therapies, derived from a plant long used in traditional Chinese medicine, have become the standard treatment for falciparum malaria. These drugs work rapidly and effectively when parasites are still susceptible, but resistance is emerging in Southeast Asia and threatening to spread to Africa. The modern face of malaria is also one of slow, grinding progress. Deaths have declined substantially over recent decades as funding for control programs increased, as net distribution expanded, as diagnostic tools improved, as surveillance systems strengthened. Countries that seemed trapped in endemic transmission have seen dramatic reductions. Some are approaching elimination, and yet the absolute numbers remain staggering. Millions of cases annually, hundreds of thousands of deaths, almost all preventable with existing tools if resources, political will, and logistical capacity aligned. Climate change adds a new dimension of uncertainty. As temperatures rise and rainfall patterns shift, the geographical range where mosquitoes can survive and where parasites can develop may expand. Highland regions in Africa that were previously too cool for year-round transmission are experiencing changes. Seasonal patterns may intensify or shift. The future distribution of malaria will depend not only on control efforts, but on environmental changes that humanity is only beginning to grapple with. In the modern world, malaria is both an ancient scourge and a contemporary failure, a disease we understand thoroughly, have tools to prevent and treat, and yet cannot eliminate from the places where it kills most readily. It persists in the gaps where healthcare infrastructure is weak, where poverty limits access to prevention, where political instability disrupts control programs, where the ecology favors intense transmission. It is a disease that reveals with brutal clarity the global disparities in health, wealth, and power. When Plasmodium parasites invade red blood cells, they initiate a cascade of events that reverberates through multiple organ systems, creating damage far beyond the immediate destruction of cells. Understanding what happens inside the body requires following the threads of pathology from the microscopic to the systemic, from the initial infection to the profound disruptions that can end in death. The blood stage of infection is where symptoms originate. As parasites multiply inside red blood cells, they consume hemoglobin, the complex protein that carries oxygen. The parasite's digestive process releases heme, the iron-containing component of hemoglobin, which is toxic to cells. The parasite crystallizes this into hemozoin, creating dark granules visible under a microscope. This is the parasite's waste, left behind when infected cells rupture. When millions of infected red blood cells burst simultaneously, they release not only new parasites but also cellular debris, hemozoin crystals, parasite proteins, and various molecules that the immune system recognizes as foreign. This debris triggers a massive inflammatory response. Immune cells throughout the body detect these molecular alarm signals and respond by releasing their own inflammatory molecules, cytokines like tumor necrosis factor and interleukins that coordinate the broader immune attack. These cytokines have multiple effects. They cause fever by resetting the body's temperature control mechanisms in the hypothalamus. The fever is not random but a deliberate immune strategy, raising body temperature to levels less favorable for the parasite. They cause muscle aches and fatigue, redirecting energy from normal activity to immune function. They increase blood vessel permeability, allowing immune cells to move from bloodstream into tissues but also contributing to fluid leakage and swelling. The destruction of red blood cells creates immediate problems. Hemoglobin released from ruptured cells breaks down into components that must be processed by the liver and kidneys. In large quantities, these breakdown products overwhelm the body's clearance mechanisms. The liver works overtime, converting hemoglobin derivatives into bilirubin, which gives skin and eyes the yellowish tint of jaundice. The kidneys try to filter and excrete these products, but in severe cases can become overloaded and fail. Anemia develops as the rate of red blood cell destruction exceeds the bone marrow's ability to produce replacements. Each red blood cell loss takes with it the oxygen-carrying capacity it provided. Tissues throughout the body receive less oxygen. The heart compensates by pumping faster and harder, but there are limits. In severe anemia, even rest requires effort. The body prioritizes vital organs, brain, heart, lungs, while peripheral tissues receive reduced blood flow. Children with severe malarial anemia may have heart rates far above normal, breathing rapidly, pale or grayish skin, and profound weakness. The spleen, positioned in the upper left abdomen, filters blood continuously, removing old or damaged cells. In malaria, it works frantically to clear infected red blood cells, parasite debris, and immune complexes. The spleen swells splenomegaly, sometimes massively, becoming palpable below the rib cage. This enlargement reflects both the increased workload and the accumulation of immune cells and debris. In chronic malaria, the spleen remains enlarged, even between infections, a physical marker of repeated disease. Plasmodium falciparum has a particularly deadly trick. It modifies infected red blood cells to make them sticky. Proteins exported by the parasite to the cell surface cause infected cells to adhere to the walls of small blood vessels, particularly capillaries and venules. This sequestration serves the parasite by keeping infected cells out of the spleen where they would be destroyed, but it creates catastrophic problems for the host. In the brain, sequestration of infected cells blocks capillaries, reducing blood flow to neurons and creating patches of hypoxia-oxygen deprivation. The endothelial cells lining brain blood vessels become activated and inflamed. Immune cells accumulate, releasing inflammatory molecules that increase capillary permeability. Fluid leaks from blood vessels into brain tissue, causing swelling. The combination of blocked blood flow, inflammation, and swelling disrupts neuronal function. Seizures can occur as electrical activity in the brain misfires. Consciousness fades as cerebral function deteriorates. Without treatment, the progression can be rapid and fatal. In the placenta during pregnancy, malaria parasites accumulate in the specialized blood spaces where maternal and fetal circulation come close together. Infected cells adhere to placental tissue, triggering inflammation that disrupts nutrient and oxygen transfer to the developing fetus. The placenta becomes infiltrated with immune cells, thickened and less efficient. Fetal growth slows. The risk of premature birth increases. Maternal anemia worsens, affecting both mother and child. The infant, if born alive, may be underweight and vulnerable to other infections. The kidneys face multiple challenges. They must filter the breakdown products of hemoglobin, process inflammatory molecules, and maintain fluid balance while blood flow patterns are disrupted. In severe malaria, acute kidney injury can develop as capillaries in the kidney become blocked with infected cells or as direct toxic effects damage filtering units. Urine production drops, waste products accumulate in blood, and electrolyte balance shifts dangerously. Metabolic disturbances compound these direct effects. Malaria parasites consume glucose from the bloodstream, and in severe infections, blood sugar can drop to dangerous levels, hypoglycemia, which further impairs brain function and can cause seizures or coma. The body's stress response releases cortisol and other hormones, but in overwhelming infection, these mechanisms can become dysregulated. Lactic acid accumulates as tissues shift to less efficient anaerobic metabolism when oxygen delivery is impaired, creating metabolic acidosis that affects enzyme function and cellular processes throughout the body. The immune system's attack on parasites is vigorous but imperfect. Antibodies bind to parasites and infected cells, marking them for destruction. Immune cells engulf and kill parasites. But the parasite is a moving target. Its surface proteins vary, changing between generations, making it difficult for the immune system to maintain effective memory responses. Even after clearing an infection, immunity is partial and temporary. Reinfection is common. The immune system learns, but slowly, incompletely, requiring repeated exposures over years to develop the partial immunity seen in adults in highly endemic regions. This partial immunity comes at a cost. The constant low-level immune activation, the repeated anemia, the ongoing demands on the spleen and bone marrow, these create chronic strain. Children who survive multiple severe malaria infections may have developmental delays, cognitive impairments, and increased vulnerability to other infections. The body's resources are continually diverted to fighting malaria, leaving less for growth, learning, and responding to other health challenges. What happens inside the body during malaria is not a single process, but a cascade parasite multiplication, triggering cell destruction, triggering immune activation, triggering inflammation, triggering organ dysfunction, each step creating problems that amplify others. The body fights back with every tool available, but against a parasite evolved over millions of years to evade, endure, and exploit, the battle is often desperate. Survival depends on whether treatment arrives in time, whether the immune system can contain the infection long enough for drugs to work, whether vital organs can maintain function through the crisis. The difference between recovery and death can turn on factors as simple as how far someone must travel to reach a health clinic, whether diagnostic tools are available, whether effective drugs are in stock. Inside every infected body, biology and circumstance contend, determining outcomes that statistics later record as cases, recoveries, or deaths. Beyond the acute fevers and immediate threat to life, malaria casts long shadows, chronic consequences that persist after parasites are cleared, burdens that accumulate over repeated infections, impacts that ripple through families and communities in patterns that become normalized yet remain devastating. In regions of intense transmission, particularly sub-Saharan Africa, children experience malaria not once, but many times during their early years. Each infection is a crisis fever, possible progression to severe disease, need for treatment, recovery period. But the cumulative effect of repeated infections creates additional layers of harm that may be less visible, but no less significant. Chronic anemia is one such consequence. Even after infections are treated, red blood cell counts may not fully recover before the next infection strikes. Children grow up in a state of persistent, mild to moderate anemia, their bodies constantly trying to rebuild blood supplies while facing repeated destruction. Anemia affects development, physical growth, slows, energy levels remain low. Cognitive development may be impaired as the brain receives less than optimal oxygen. Studies in highly endemic regions show associations between repeated malaria in early childhood and lower educational achievement, delayed motor development, and reduced height and weight compared to peers with less exposure. The immune system, constantly engaged in fighting malaria, has less capacity to respond to other infections. Children in malaria-endemic regions face multiple infectious disease threats simultaneously: bacterial pneumonia, diarrheal diseases, measles, other tropical illnesses. Malaria makes all of them more dangerous. The interaction works both ways. Other infections can trigger malaria relapses, worsen malaria severity, and complicate treatment. This syndemic, overlapping epidemics that interact creates health burdens greater than the sum of individual diseases. Pregnant women face particular long-term consequences. Each pregnancy brings increased vulnerability to malaria as the placenta provides a new site for parasite accumulation. Women who have developed partial immunity through years of exposure can lose that immunity during pregnancy, becoming susceptible to severe disease. Malaria during pregnancy increases risks of maternal anemia, miscarriage, stillbirth, premature delivery, and low birth weight. Babies born underweight face higher mortality in their first months of life and increased risk of developmental delays. A mother who survives severe malaria may face prolonged recovery, affecting her ability to care for newborns and other children. The neurological consequences of cerebral malaria can be permanent. Children who survive comas may wake with brain damage, motor impairments affecting movement, epilepsy causing ongoing seizures, cognitive deficits affecting learning and memory, behavioral changes. Families watch children who were healthy and active become disabled, requiring care that poor rural families are often ill-equipped to provide. Educational opportunities disappear. The child's entire life trajectory is altered, and the family bears the burden, emotional, physical, economic, without support systems adequate to the challenge. Economically, Malaria perpetuates poverty through multiple mechanisms. Direct costs include money spent on treatment, transportation to health facilities, and diagnostic tests. Indirect costs include lost productivity when people are too sick to work or must stay home to care for sick children. In agricultural communities where survival depends on physical labor, a week or two of illness during planting or harvest seasons can mean failed crops and food insecurity. Adults with repeated malaria infections work fewer days, earn less income, save less for emergencies. At the community level, endemic malaria affects economic development broadly. Businesses locate elsewhere when workers are frequently ill. Agricultural productivity is lower in malaria zones. Tourism, which could bring income to tropical regions, is discouraged by disease risk. Foreign investment is reduced. Areas with intense malaria transmission often remain economically marginalized, creating a vicious cycle where poverty prevents implementation of control measures and ongoing transmission perpetuates poverty. Educational systems suffer directly. Children miss school repeatedly during infections. Teachers are frequently ill. Schools in remote areas struggle to attract staff willing to work in high transmission zones. The cognitive effects of repeated malaria in early childhood reduce learning capacity. By the time children reach school age, they may already be behind peers from less endemic regions. Dropout rates are higher, literacy rates lower. The human capital that drives economic development is systematically eroded. The mental health impacts are less documented, but no less real. Parents who have lost children to malaria carry grief that communities may normalize, but individuals still feel acutely. The stress of living with constant disease threat, of watching children suffer repeatedly, of fearing each fever might progress to something deadly. This chronic stress affects mental well-being. Caregivers, usually women, experience exhaustion not just from repeated episodes of nursing sick children, but from the relentless vigilance required in high transmission settings. At the global scale, the burden is almost incomprehensibly large. Hundreds of millions of cases occur annually. Even as deaths have declined due to control efforts, the sheer number of people experiencing illness, missing work and school, requiring treatment, remains staggering. The economic costs run to tens of billions of dollars in direct healthcare expenditure and lost productivity. But these numbers capture only part of the burden. They cannot quantify pain, fear, grief, lost potential, or the ways malaria constrains life choices and possibilities. Malaria also interacts with other major global health challenges in ways that amplify burdens. HIV and malaria affect overlapping populations in sub-Saharan Africa, and each makes the other more dangerous. Malnutrition and malaria interact. Malnutrition increases malaria severity, and malaria worsens nutritional status by causing appetite loss, nutrient malabsorption, and increased metabolic demands. The Sustainable Development Goals' international targets for improving health, education, and economic well-being are harder to achieve in regions where malaria remains endemic. Some populations face additional vulnerabilities. Refugees and displaced populations, living in temporary camps with poor sanitation and limited healthcare access, experience high transmission rates and limited access to prevention or treatment. Migrant workers moving between regions may lose partial immunity when they leave endemic areas and face severe disease upon return. Indigenous populations in remote areas may lack access to health services entirely. Marginalized groups already facing discrimination find those inequities amplified by disease burdens that healthcare systems fail to address adequately. The long-term consequences of malaria are not distributed randomly but follow fault lines of inequality affecting the poor more than the rich, rural populations more than urban, children more than adults, women during pregnancy, the already marginalized. The disease entrenches disparities, making escape from poverty harder, making educational achievement less likely, making economic development slower. It is a disease that, left unchecked, becomes woven into the fabric of societies, affecting everything from family structure to agricultural practices, to settlement patterns, to political stability. Yet the burden is not inevitable or unalterable. Where control programs have been sustained and well-resourced, malaria has been pushed back dramatically. Countries have moved from endemic transmission to elimination. Deaths have declined even in areas of intense transmission as bed nets, diagnostic tests, and effective treatments become more widely available. The burden is large because malaria is a formidable foe, but it is reducible, controllable, and in many settings, eliminable. The question is not whether the tools exist, but whether the political will, funding, and logistical capacity can be mobilized consistently enough, over long enough periods, to break transmission cycles and prevent their re-establishment. The long-term burden of malaria ultimately reflects not just biological reality, but human choices about resource allocation, equity, and commitment to health as a global public good. To understand malaria fully is to see it not as an isolated medical problem, but as a thread woven through the entire tapestry of human history, shaping and shaped by our species' journey from small populations in African forests to a global civilization of billions. The relationship between humans and malaria is evolutionary, ecological, historical, economic, and ultimately existential, a story about how we live with microscopic predators, how we adapt to threats, and how we choose to protect the vulnerable among us. The evolutionary dimension is profound. Malaria has been such a powerful selective force on human populations that our genes bear its imprint. In regions with long histories of intense falciparum malaria, genetic mutations that would otherwise be harmful have spread through populations because they provide protection against the parasite. The sickle cell trait, a mutation in the hemoglobin gene, causes severe disease when inherited from both parents, but provides significant protection against severe malaria when inherited from only one parent. In malaria-endemic regions, this mutation is common. In regions without malaria, it is rare. Similar patterns appear with thalassemia, G6, PD deficiency, and other genetic variants. Human populations have been shaped by malaria, carrying in our DNA the marks of thousands of generations of lethal selection pressure. This evolutionary legacy raises uncomfortable questions. Genetic variants that protect against malaria can cause other health problems. Sickle cell disease, when someone inherits the mutation from both parents, is a serious, chronic condition. The distribution of these protective mutations maps precisely onto historical malaria zones, demonstrating how deeply the disease has influenced human genetic diversity. Our bodies are in some sense compromises, adapted to survive in environments where malaria was a constant threat, even at the cost of other vulnerabilities. Ecologically, malaria sits at the intersection of climate, landscape, human settlement, and vector biology. The disease cannot exist without mosquitoes, and mosquitoes cannot thrive without specific environmental conditions. Human activities reshape those conditions constantly. Deforestation creates new pools and puddles. Irrigation agriculture creates ideal breeding sites. Urbanization can either increase transmission by creating standing water in crowded settlements, or decrease it by paving over breeding sites and improving drainage. Dam construction changes river flows and creates reservoirs where mosquitoes breed. Every human intervention in the landscape alters the equation of transmission, usually in ways that become apparent only after new disease patterns emerge. Climate change introduces radical uncertainty. As global temperatures rise, the geographical range where malaria can be transmitted may expand. Highland regions in Africa and South America that were previously too cool for mosquitoes or parasites may become suitable. Temperature affects not just mosquito survival, but also how quickly parasites develop inside mosquitoes. Warmer means faster development, means more infectious. Mosquitoes. Rainfall patterns influence breeding sites. Extreme weather events create new habitats or destroy control infrastructure. The future geography of malaria will be written partly by carbon emissions and climate policy, linking the disease to the largest environmental challenges humanity faces. Anthropologically, malaria has shaped human culture and social organization in countless ways. Traditional practices for managing fever, beliefs about disease causation, settlement patterns prioritizing locations away from swamps, seasonal migration patterns avoiding peak transmission times all reflect generations of lived experience with the disease. Indigenous knowledge systems developed sophisticated understandings of environmental factors affecting illness, even without microscopes or knowledge of parasites. The cinchona tree in South America, source of quinine, was used by indigenous peoples long before European colonizers learned of its anti-malarial properties. Traditional medicine systems worldwide include preparations claimed to treat fever, some of which likely had real though limited efficacy against malaria. Scientifically, understanding malaria required centuries of careful observation, hypothesis testing, and technological development. The parasite was first observed in blood by French physician Charles Laveran in the late 19th century, work that earned him a Nobel Prize. British physician Ronald Ross demonstrated in the late 1800s that mosquitoes transmitted the parasite, another Nobel-worthy discovery. The complex life cycle involving liver stages, multiple blood stages, and sexual reproduction in mosquitoes was pieced together through painstaking microscopy and experimentation. Each advance opened new possibilities for control. You cannot fight what you do not understand. The development of antimalarial drugs traces a path through chemistry, pharmacology, and evolving parasite resistance. Quinine from cinchona bark was used for centuries. Chloroquine, synthesized in the mid-20th century, seemed like a miracle drug, inexpensive, effective, relatively safe. But resistance emerged within years in some regions, spreading globally over decades. Each subsequent drug, mefloquine, sulfadoxine, pyrimethamine, artemisinin and derivatives, followed similar trajectories. Initial efficacy, widening use, eventual resistance. The biological arms race between drug development and parasite evolution continues, a reminder that solutions are temporary, that parasites adapt, that vigilance cannot lapse. Vaccine development has proven extraordinarily challenging. Unlike viruses or bacteria, parasites are complex organisms with thousands of genes, elaborate life cycles, and sophisticated immune evasion strategies. The parasite's ability to vary its surface proteins makes it difficult for antibodies to recognize and attack. Decades of research produced many failed vaccine candidates. Recently, a vaccine showing partial efficacy in young children has been approved and is being rolled out in endemic regions. Not perfect, not a silver bullet, but a new tool that may save many lives when combined with other interventions. Public health approaches to malaria control have evolved through trial, error, and hard-won experience. Early campaigns focused on eliminating mosquitoes through environmental modification, draining swamps, applying oil to water surfaces, stocking ponds with fish that eat mosquito larvae. The DDT era brought powerful insecticides that could reduce mosquito populations dramatically, leading to successful elimination campaigns in many countries. But resistance developed. Environmental concerns about persistent pesticides grew, and the approach proved unsustainable in regions of intense transmission. Modern integrated approaches combine multiple interventions: insecticide-treated bed nets for personal protection, indoor spraying in some settings, rapid diagnostic tests for accurate case detection, artemisinin-based combination therapy for treatment. intermittent preventive treatment for pregnant women and infants, environmental management where feasible, surveillance systems to track cases and guide responses. No single intervention is sufficient. Success requires sustained implementation of combinations tailored to local contexts, funded adequately, and maintained over years. The malaria story is also about global solidarity and inequality. The disease was eliminated from wealthy countries, first Europe, North America, parts of Asia, where resources allowed comprehensive control programs. It persists most intensely in the poorest countries, particularly in sub-Saharan Africa, where healthcare systems are weakest, where resources are most limited, where the ecology most favors transmission. Billions of dollars in international aid flow annually toward malaria control, funded by governments, private foundations, and international organizations. These resources have achieved real impact, millions of deaths prevented, hundreds of millions of cases averted. Yet funding is always insufficient relative to need, always vulnerable to political changes, always requiring advocacy to maintain. Technological innovations continue to emerge. Genetic modification of mosquitoes to make them resistant to parasites or unable to reproduce offers potential future tools. Gene drives genetic modifications that spread rapidly through mosquito populations could theoretically crash mosquito numbers or eliminate their capacity to transmit. Parasites, though ecological and ethical concerns require careful consideration. Better diagnostic tools, including rapid tests usable in remote areas without laboratories, improve case detection. Smartphone apps aid surveillance. Artificial intelligence helps optimize resource allocation and predict outbreaks. The toolkit expands, but deployment remains the challenge. The question of elimination versus control, whether to aim for complete interruption of transmission or sustained reduction of burden, animates ongoing debates. Elimination requires intense effort, sustained funding, strong surveillance, and ability to detect and treat every case. In low transmission settings, elimination is achievable and has been achieved. In high transmission settings, particularly parts of Africa, elimination remains beyond current capacity. The pragmatic approach focuses on burden reduction, preventing deaths, reducing severe disease while building capacity toward eventual elimination as resources and tools improve. Looking back to the forests where malaria originated, the full arc becomes visible. A parasite that evolved to exploit primates found in humans a host that would carry it across the world, creating opportunities for spread on a scale no forest ecosystem could support. Human population growth, agricultural intensification, global trade, and urbanization created environments where transmission could explode. But humans also developed science to understand the disease, public health systems to fight it, technologies to prevent and treat it, and the capacity for collective action that has pushed malaria back from much of the world. The larger human story of malaria is unfinished. Millions still fall ill annually. Hundreds of thousands still die, mostly children who never had the chance to live full lives. The tools exist to change this, to reduce malaria burden dramatically, to eliminate transmission in region after region, eventually to confine the parasite to tiny pockets of remaining transmission that could, with sustained effort, be extinguished entirely. But tools alone are insufficient. Success requires resources, political commitment, international cooperation, healthcare infrastructure, strengthening, and sustained effort over generations. Malaria is a mirror held up to humanity, reflecting both our vulnerabilities and our capabilities. It shows how we remain embedded in natural ecosystems, subject to the same evolutionary forces as other species, vulnerable to microscopic parasites we can barely see. But it also shows our unique abilities to understand complex biology, to develop sophisticated interventions, to organize collective responses, to choose to protect the vulnerable, to persist in the face of setbacks. The story of malaria is ultimately about whether we can mobilize those capabilities consistently enough, equitably enough, and sustainably enough to protect all people, not just those fortunate enough to live where the disease has already been defeated. The forests where it began still exist. The mosquitoes still fly. The parasites still circulate, but the outcome is not predetermined. The next chapters of this ancient story will be written by human choices about funding priorities, research investments, healthcare infrastructure, global cooperation, and commitment to the principle that every life, regardless of geography or circumstance, deserves protection from preventable suffering and death. Malaria has shaped human history. The question now is whether humanity can reshape the future of malaria, bending the trajectory from endemic threat toward elimination, from millions of cases toward zero. The biological foundations are ancient and enduring, but the human story continues to evolve, and in that evolution lies the possibility of change.